Well, again, my dear students, we are here today again with the subject of physiology of lactation. See, before we proceed with our today's lecture, as usual, let's seek the guidance to the righteous from the Almighty, and also seek from Him the blessings and the knowledge that's beneficial to the mankind. Above all, we are here to acquire some knowledge, and of course, this one knowledge should be beneficial to the mankind. That should be our goal, that should be our motive, that should be our objective. Well, dear students, today we'll be discussing about synthesis of milk in mammary glands. We have discussed a lot about the mammary glands, how they develop from the very beginning when the baby or the female is in the womb itself. Then through different stages of its life, be postnatal, postpubertal, during pregnancy, and then how this mammary glands, they get developed and the milk producing capability and ability is being acquired during that last stage of pregnancy and after the parturition. And we have discussed about the lactational curves. Now, dear students, over here, the mammary glands too produce milk. And the milk is what we have already discussed, that is an elixir, a complete food. And the whole growth of the babies depends upon the milk for a certain period of their early life that supports the wholesome development of the baby, the newborn. So this milk basically does contain all those constituents which are required for the development of body or the development of an organism or an animal or a newborn. So major classes of those constituents we know in the food or the diet. It should contain the basics of energy, proteins, lipids, which produce, the raw material for energy, cellular growth. And besides that, you need vitamins, you need minerals. So this milk does contain all those things. The source of energy as lactose, the proteins as casein, the fats or the lipids. Besides that, a lot of minerals, a lot of vitamins, and even the immunoglobulins. That is a specialty with the milk, that it does contain the immunoglobulins, particularly in the colostrum, which is rich in the immunoglobulins that provides the immunity to the newborn. So we'll see that how these milk constituents are being produced by the mammary gland. What are the raw materials for it, where from they are getting derived, and does mammary gland take any role in its, formation, in its synthesis? We'll be discussing that today. So the milk components are primarily derived from the blood. The raw materials are taken from the blood supplying mammary gland. So the mammary glands are being well supplied with the blood, and all those constituents of the milk, their source is from blood. The raw material comes from the blood. It gets modified, it gets somehow say, process it in the mammary gland and produces the energy source, the protein source, the lipid source. So material taken from the blood by the epithelial cells of the gland at its basal membrane are either transported to the lumen of these epithelial cells through apical membrane as such, or may enter into the epithelial cell machinery to modify it, synthesize it, or package it, and then pass on to the lumen. So blood supply is on the basal membrane of these epithelial cells. So epithelial cells from the blood, generally all those things in the blood, they come into the extracellular fluid. From the extracellular fluid, the cells take it through the membranes and through different say mechanism, maybe active transport, maybe through diffusions and many other routes. So once it comes into the epithelial cells, it may be taken to the lumen, or you can say into the milk itself as such, or it may be processed in the cell or epithelial cell, or maybe modified, maybe synthesized, maybe packaged. It will depend upon the constituent of the milk. We'll be discussing later on. In the dairy cow, energy requirements for lactation can reach 80% of the net energy from intake, and lactose production can utilize 85% of the circulating glucose. So lactose, which is basically the major energy content or major carbohydrate in the milk, that is being produced from the blood in the glucose, and 85% of it is glucose, which is there in the blood being supplied to the mammary glands, the blood that's being utilized in the production of the lactose. To conserve glucose for lactose synthesis, ruminants have developed glucose sparing mechanisms in the gland and other body tissues to preferentially use other metabolites such as NEFAs and the ketone bodies as energy sources for metabolic activities, sparing the glucose for synthesis of lactose by the mammary glands. So lactose is the major carbohydrate component of the milk, which is especially in the milk. You won't find lactose in the body, you won't find lactose anywhere except in the milk. So during lactation, the components of the milk are secreted across the mammary epithelium and or into the lumen by one of the 5 routes. So 5 routes are being taken by the mammary gland to either synthesize or to pass on those constituents from the blood into the lumen or into the milk via epithelial cells. So one is the membranal route, means through membrane, or you can say transmembrane. And then Golgi route, the constituents reach to the Golgi, get packaged there, and then get secreted on the luminar side of the epithelial cells. Milk fat route is again one of the route that milk fat takes through processing it, making those globules, milk globulets or milk droplets. Then transcytosis and paracellular routes. These are the 5 routes through which the constituents pass on from blood to the lumen of the alveoli, or you can say into the milk. So interstitial fluid derived substances like water, urea, glucose, ions cross the basolateral membrane of the epithelial cells, traverse the cytoplasm, and diffuse across the apical membrane into the alveolar lumen. That is a membrane transport. Simply, these all things, they will enter, they will cross the basolateral membranes, maybe balanced by, say, concentration gradients, or maybe taken some special pumps, and they get inside the cytoplasm of the cell and simply diffuse across the epithelial membrane into the alveolar lumen, or other words, into the milk. So that's a membrane route. Then products that utilize the Golgi route get first synthesized within the cell and then packaged into the secretory vesicles, but from the stakes of this Golgi membrane, travel to the apical membrane, fuse with the membrane, and release their contents into the lumen, what we otherwise name as exocytosis. So many of these constituents of the milk, they come into the cytoplasm of these epithelial cells, then get processed into the Golgi, packaged there. So cut off from those Golgi membranes, then make those secretory granules or secretory vesicles, you may call it. And those secretory vesicles will travel to the apical membrane, it will fuse there, and then they will push these contents into the lumen, or in other words, into the milk. So that's the process of exocytosis. Similarly, in milk fat route, this milk fat which is being produced by the epithelial cells of the mammary gland and comes into the milk. So it takes a route in which a portion of the apical membrane surrounds the droplet and buds through the apical membrane. Portions of the cytoplasm and other cellular substances can be trapped in the milk fat droplet. That's the lipid soluble drugs and the hormones. So here it's not the exocytosis basically. So the fat globules, they are membranes, or they are, say, trapped inside the membrane, and then these membranes get pinched off from the apical surfaces, apical membrane, into the milk. And during this process, they take other things with it also. And it's not simply the exocytosis, but it is the budding of these, say, or pinching of globules along with the membrane as a milk droplets, fat droplets into the milk. Then trancytosis, transport, that is, vesicles derived from the basolateral membrane via endocytosis or pinocytosis migrate across the cellular cytoplasm and secreted into the milk via exocytosis. E.g., for example, immunoglobulins and albumin, they are as such taken in the mammary gland cells or the epithelial cells by a process what we name as pinocytosis or endocytosis. And then they come into the cytoplasm of these cells as those vesicles, membrane-bound vesicles, and then they reach to the apical membranes, and through exocytosis, once they reach to the apical membrane, fuse with the membrane, and then push, release the materials, the immunoglobulins, the albumin, in certain stages of the lactation into the milk. That's transcytosis, transport of things like immunoglobulins, albumins. Other is a paracellular transport, materials from the interstitial fluid that's there on the basolateral membrane of these apical cells, epithelial cells, between the adjacent cells and into the milk takes place. So during this colostrogenesis, colostrum, when it's being produced, much of the immunoglobulins and the other serum-derived proteins move into the mammary secretions via the paracellular route prior to the formation of tight junction. So its paracellular transport is between those cells, between the cells in the epithelial cells of a mammary gland. There are tight junctions, and if they are being, say, somewhat compromised towards the early lactation or towards, just after parturition, these tight junctions are permeable, and therefore many things besides water and ions, minerals, even the immunoglobulins do cross through these tight junctions on the other side of the membrane. That's what we name as paracellular route. So these are the 5 types of the routes which the constituents of the milk take while crossing from basolateral membrane to the apical membrane into the milk. So you will find here in a picture, you will see this is a one, they have the tight junctions over here, right? So this is what you name as, this first is exocytosis. You will find that from the Golgi, you will find these secretory vesicles, they will fuse with the membrane and will push the contents into the luminar side, into the milk. This is here the basal membrane, and this is the capillary blood supplying over here. So in the blood, everything from the capillary will come into the extracellular fluid. From here, they will enter through these basolateral membrane. This is what we name as basal membrane over here. This is a basement membrane, and it will come over here. So this will be taken one route, other route, the fats, which will take the route in a fat globule that will take the membrane along with of this apical membrane and will be pushed here as this milk fat droplet, and see, along with the membrane, along with these fats as well as many other things. Third will be simply a transmembrane. You see here is apical transport. The things will come from here and they will pass directly from here to here. And fourth will be taken by what we name as transcytosis, means the immunoglobulins particularly, they will come over here, they will form that invagination, then a vesicle, and that will bud off, that will pinch off, or what you name as pinocytosis, endocytosis. It will come here into the basilar membrane, into this apical membrane, and here by the process of exocytosis, it will push those constituents, or you can say immunoglobulins, maybe albumins, to the milk itself. And then fifthly, what we name also as paracellular. The things will come through these tight junctions because this is permeable. These intercellular junctions, they are over here, say, open or permeable to many things. So things will come to this side and maybe sometimes to this side also. So this is a general view of these, how these milk constituents, they get crossed through epithelial cells into the milk or into the lumen. See now, this is one more picture over here. You will find again, these are the tight junctions and many things will cross these. You see salts, leukocytes, they will cross through these paracellular pathway, right? And you'll see similarly, these are you see mitochondria, this rough endoplasmic reticulum, this is Golgi, and this is secretory vesicle, as I have discussed, this exocytosis over here. You will find the glucose, they will come over here, they will perform, and they will be processed, they will be packaged as the lactose over here, and then will be in a secretory vesicle and will be pushed out here. And similarly, even proteins you will find in a smooth endoplasmic reticulum. This was a rough endoplasmic reticulum. This is a smooth endoplasmic reticulum. Fatty acids, acetate, beta hydroxybutyrate, even glycerol, they all will be processed over here and they will form the droplets, fat droplets, and ultimately for droplets, they will pitch out of this luminar membrane or apical membrane into the milk as a milk droplets. Or you can see simply that immunoglobulins, they will see here, they will form say through endocytosis, they form the endocyte, or they will form that vesicle and that get pinched off from this apical, rather this basolateral membrane, and will take to the apical membrane, and here they will fuse with this apical membrane and then push the contents, particularly the immunoglobulins. So these are say many constituents of the blood. You will see they take different routes. Salts, leukocytes will take the paracellular route, amino acids, they will take the rough endoplasmic route, and then Golgi and the secretory vesicles. Similarly, the galactose or the glucose, like lactose, they will take the same route as secretory vesicles, whereas fatty acids, butyric, beta hydroxybutyrate, glycerol, they will take smooth endoplasmic reticulum and fat droplet route. Whereas immunoglobulins, they will take this paracellular this pathway, or we have also transcytosis, or you can say transcytosis pathway. It will take the pathway. So moving further, we'll see now the different constituents, how they are being produced. If we take the lactose, as I have already discussed, lactose is one of the major carbohydrate in the milk, which is a disaccharide made up of two monosaccharides, the glucose and galactose. You won't find anywhere in the body of an animal this lactose, except maybe somewhere when there is any disturbance in those tight junctions, when they get permeable, and some lactose may leak into the blood. Maybe during disease conditions and maybe very early lactations. And this is present in the milk of most mammals and is least variable constituent of the milk. So this carbohydrate, moiety of the carbohydrate constituent of the milk, that what we name as lactose, that's in most of the mammals least variable constituent. It doesn't vary much, rather ranges between 4 to 5% most of the domestic animals, and it is highest in the humans, some 7%, whereas very low in the sea mammals, maybe 1% or even less than 1% of this lactose. You see, lactose is very you can say osmotic molecule. It draws this water. It's a strong osmotic molecule, a ready digestible source of glucose for the neonates. And this is the lactose which gives energy to the neonates, to the newborns. And this lactose is being digested by enzyme, what we name as lactase, that's being present in the neonates in the early life. And that's unique to the mammary gland only, that I have discussed, and you won't find this anywhere in the animals, in any other organs. And the major osmoregulatory regulator of the milk. Glucose passes across the Golgi membrane into the Golgi lumen by a glucose transporter, that's GLUT1. The presence of GLUT1 on the Golgi membrane is specific to the membrane epithelial cells. So this glucose enters into the epithelial cells through simply as it is being diffused as in the other cells, as per this utilization, as per its concentration gradient, or maybe through these glucose transporters. Once it is in the cytoplasm of the cell, it is taken into the Golgi membrane, and into the Golgi membrane is taken by a transporter, that's GLUT1. And this GLUT1 is only present in the Golgi membranes and is specific to the mammary epithelial cells. Then you'll find that once this glucose comes into the membrane, it has to be fixed there because it should not get leaked out. So UDP galactose is actively transported into the Golgi lumen, which is a rate limiting here. And this UDP, that's uridine diphosphate glucose, is not transported into the Golgi. Okay, this lactose is non-permeable disaccharide and cannot diffuse out of the Golgi membrane or out of the secretory vesicles. So once this lactose is formed from the glucose, you see, in other words, when glucose enters into the cells, it is phosphorylized, and then because of that phosphorylation, this glucose doesn't leak out of the cells, and then it enters into the different pathways for energy release, maybe that pathway, maybe TCA cycle, maybe those this pathway where they are utilized for the energy production. Here, this UDP is rapidly hydrolyzed to the UMP and inorganic phosphate by the nucleosides. So once this UDP generated from the lactose synthesis could be inhibitory to the lactose synthesis if it accumulated in the Golgi. So UDP is, if it gets, say, once UDP is there, uridine diphosphate, so it doesn't say permit the glucose to leak out of the Golgi. So, or lactose, it's being trapped in the Golgi for further processing. And if UDP remains there, so it becomes an inhibitor to the lactose synthesis. So this UDP is to be processed, it is to be hydrolyzed to UMP, and then UMP is actively removed from the Golgi, and in organic phosphate which comes from this, that diffuses out of it, and then lactose synthesis reaction is one way. Say once lactose is being formed from the glucose and galactose, it is not say hydrolyzed to form the glucose and the galactose. That is, it is a one-way reaction. So lactose synthase enzyme, it's composed of two subunits. This lactose, which is formed from glucose and a galactose, it requires two subunits, or it requires the different classes of the animal, these enzymes. One of the enzyme, that's the lactose synthase enzyme, that has two subunits. One, that's a galactosyl transferase, found in several tissues only on inner surface of the Golgi, and then very important, alpha-lactalbumin specifically catalyzes transfer of galactose to the glucose. Then this lactalbumin is only present in the mammary glands. It's a protein which is there present, being synthesized by the mammary glands itself. So lactose and alpha-lactalbumin are so co-secreted via Golgi secreted vesicles. So once this lactose is being formed, since we require for its formation the lactalbumin, alpha-lactalbumin. So once these lactose are packaged into secretory vesicles, it's along with alpha-lactalbumin. So lactose along with the alpha-lactalbumin, they are co-secreted via Golgi secretory vesicles. So alpha-lactalbumin is limiting to lactose synthesis. So there are some reports, if you have the strategies to increase alpha-lactalbumin, probably you may increase the milk secretion, milk yield. Then we'll find that, see here, we find that glucose is the major, source of this lactose, this glucose, right over here. We'll see here, otherwise in the normal conditions, this glucose through a hexakinase, this hexakinase is put into the glucose 6-phosphate as phosphorylated, so it does not come or leak out of the cells, it will remain there. And then phosphoglucomerase, or you can say glutase somewhere, glucose 1-phosphatase, then UTP glucose through UDP glucose, that's one enzyme. That's a pyrophosphorylase, very important, that converts the glucose 1-phosphate into the UTP glucose. Then UTP glucose to UDP galactose, that's the one important enzyme that what we name as epimerase, or UDP glucose epimerase. So it converts this UTP glucose into the UDP galactose, and this inside this Golgi, along with the glucose, forms the lactose under the presence of one important enzyme, that's the galactosyl transferase and alpha-lactalbumin, together called as lactose synthase. So this is, you'll find here, it's the cytoplasm, this is the Golgi. So in the cytoplasm, it will come glucose, it will be fixed there through hexokinases, and then change to the glucose 1-phosphate by this phosphoglucomatase, and then UDP glucose by this phosphorylase, and then to the UDP galactose by one epimerase, and then this UDP galactose will enter into the Golgi through this transporter, UDP galactose transporter, and here the glucose through glucose transporter GLUT1 into the Golgi. Glucose will come, and then galactose, UDP galactose will come, and both will combine together, form a lactose. There's a molecule of glucose and one galactose will form a lactose. There's a disaccharide under the influence of that lactase or lactose synthase, which has two components, the galactosyl transferase and alpha-lactalbumin. So this is how this glucose is being, this lactose is being produced in the mammary cells. And once into the Golgi, they get packaged into secretory vesicles, then separate from these Golgi as secretory vesicles into cytoplasm, or reach to the apical membrane of these mammary cells or mammary epithelial cells, and then through the process of exocytosis, push the lactose into the milk or into the lumen, in other words, along with the lactalbumin, or you say alpha-lactalbumin. This is how this lactose is being produced. You'll find here these dietary NDF or NFSC, neutral detergent fiber or non-fibrous carbohydrates. In case of the ruminants, they will come into the lumen. If some glucose which escapes the ruminal fermentation, it will come into the intestine. Most of it will be processed here, and propionate will feed different fatty acids are being produced. VFAs are being produced in the rumen. One of the important is propionate. That's the source of the glucose in the ruminants. It will come into the liver, and propionate, it will produce a glucose, and glucose will come into the circulation. From circulation, it will go to the mammary gland, and in the mammary gland, it will be produced with glucose and a galactose. Glucose and galactose, they will combine to form lactose, and you will get lactose into the milk. This is how the origin of lactose is there, how this lactose, which is not found in the body but found only in the milk, is secreted by the mammary gland epithelial cells out of the glucose, which is the source of its, or you can say, the raw material for the production of lactose, and under the influence of different enzyme systems in the Golgi. In the Golgi, it is being processed, packaged, and ultimately reaches to the outside or into the milk, or being secreted into the milk through exocytosis. This is about the lactose. Next lecture will find other constituents of the milk. This time we will take leave over here. With this, hope that you have understood how this lactose is being produced by the mammary glands. Thank you. Thanks a lot. We'll be meeting again. So the synthesis of milk, which one in this synthesis of fats in the milk, one more constituent, very important constituent, very important energy source in the milk, that is fats, the milk lipids. We already discussed about the lactose, which was a very important carbohydrate source in milk, and now one more, that's this milk fat or the milk lipids. You see, milk fat is primarily composed of triglycerides that are composed of variable mixtures of fatty acids and glycerol. And milk fat triglycerides, 90% of total milk lipids. So these milk triglycerides or milk fat triglycerides, they constitute 98% of the total milk lipids, and most of it contains saturated fatty acids. That means, saturated maybe from C4 to carbon 8, maybe from small fatty acids to very long fatty acids. And most of them, it's the saturated fatty acids, or maybe very little of unsaturated palmitoic acid, maybe oleic acid, maybe linoleic acid. So the source of these fatty acids are two. There are two important sources of fatty acids in case of mammary gland from which these malic fat is being produced. One is that blood plasma lipids. Since blood does contain the lipids, dietary lipids, fatty acids, they compose 50%. That means what the animal eats and the lipids it absorbs from the intestine does come into the plasma, and this blood circulates into the mammary gland. Mammary gland draws it and produces the fat in the milk from it, and it may be 50% may be contributed by this say blood plasma lipids. And among that 50% that is being produced, that is being contributed by the blood plasma lipids, dietary lipids will constitute 80%, and adipose derived will be say providing only 20%. So it will be only mostly the dietary lipids, and from the adipose very little, only 20%. And most of it will be say which comes from blood plasma lipids, that will be C18 means long chain fatty acids, and C16. C18 100% very saturated, and then C16 again a long 70 to 80% of this milk fat in the milk fat that will contain these saturated fatty acids. Then one more pathway of this production of lipids into the milk is de novo synthesis. That is, mammary gland itself synthesizes the lipids from the epithelial cells of these mammary glands, and that constitutes 50%. That means 50% of these lipids, one is being provided from the blood plasma lipids, and the 50% is being provided by the de novo synthesis, or the itself mammary gland synthesis, 50%, and that 50% is constitutes mostly of small or medium fatty chain fatty acids, C4 to C10, 100%. Most of these are small and medium chain fatty acids which are being produced by de novo synthesis, whereas which come from the blood or from plasma lipids, that is mostly say long chain fatty acids, C18, even more C18 to C60. Dietary lipids are mostly saturated long chain fatty acids, and hydrogenation converts unsaturated fatty acids to the saturated. So whatsoever the dietary those fats, they get consumed by the ruminants. Since in the rumen itself they are processed, they are hydrogenated, and all of them are being converted into saturated fatty acids, saturated long fatty chain acids, and doesn't remain unsaturated. So most of that fats which are being derived from these plasma lipids, they are say saturated fatty acids. Only the portion which is being produced by the mammary gland itself through de novo synthesis will find small chain fatty acids, not long chain, and maybe some unsaturated too. In de novo, complete elongation is up to 16, C16 and not beyond that. So the maximum length of that fatty acids will be C10, which are being produced by the de novo synthesis. Whereas de novo, mostly short chain or medium chain fatty acids are produced in the cytoplasm of the mammary epithelium. The source for de novo synthesis is beta hydroxybutyrate and acetate. So for this de novo synthesis, it doesn't take it from the plasma lipids, but it take it as beta hydroxybutyrate and acetates that are being produced in the ruminants through this ruminal digestion, and volatile fatty acids come into the liver and then to the mammary gland. And these butyrates and acetates are being utilized by the mammary epithelial cells for de novo synthesis of fatty acids through acyl-CoA synthetase and acetyl-CoA carboxylase and fatty acid synthase. These are three important enzyme systems which are being utilized in the de novo synthesis of fatty acids from butyrate and from butyrates and acetates. And it produces small, or you can say short to medium chain fatty acids. The enzyme that we have discussed, that's the acyl-CoA synthetase, is activator of acetic acid and hydroxybutyric acid into these amices, which are transformed to the acetyl-CoA after connecting to pyruvate. So these then ACC enzymes, or in other words, what you name acetyl-CoA carboxylase, that system for the formation of malonyl CoA MCFAs, medium chain fatty acids, or short chain fatty acids by this synthase, fatty acid synthase action in the cytosol. It's all happening in the cytosol. Other enzymes such as ATP citrate, lysase, or fatty acid elongase, and the steroid CoA desaturase have a significant role in the de novo synthesis. There are three important those enzyme systems which I have discussed. That's the acyl-CoA synthetase, then acetyl-CoA carboxylase, and then fatty acid synthase. These are very important primary enzyme systems which are being taken into by epithelial cells in the production of fatty acids from butyrates and acetates. So dietary strategies can modify the milk fat composition of the lactating ruminants. Dietary supplementation with the non-conventional feed setups rich in unsaturated fatty acids have an impact on milk fortification with PUFAs. There has been a study, many studies have been there, and they have demonstrated that in case of the ruminants, if you are providing or you are your dietary supplementation is with some unsaturated fatty acids, the food steps which are rich in unsaturated fatty acids, then you can expect milk with those PUFAs, or you can say polyunsaturated fatty acids. It's a good thing. And presently we are very cautious about the milk fat, which otherwise is very much saturated, and if I make it unsaturated fatty acids more available in the milk, it can be fortified, it can be liked by the public, and it is probably very much in the demand. The other sources of fatty acids taken up by the mammary gland is blood plasma coming from the intestinal absorption of the dietary and the microbial fatty acids. Long chain fatty acids, say C18, along with 50% C16 and a small portion of C14, they get derived from the blood plasma lipids. Small portion is released by the lipoprotein lipase. You see, one important lipoprotein lipase is one of the important enzymes which produces or releases these fatty acids from the triglycerides that are circulating as chylomicrons, or very low density lipoproteins, VLDLs, or from the plasma NEFA bound to the albumin. So these can be the sources of the lipid in the milk, which are being produced, which are being released by a lipoprotein lipase, an enzyme. This enzyme acts on chylomicrons which are circulating there, or even low density lipoproteins, or even the plasma NEFA bound to the albumin. So in non-ruminant species, metabolism of the glucose by the epithelial cells provides the acetyl-CoA, which is used as the carbon primer for fatty acid synthesis. Since in case of non-ruminants, you won't find the acetates and the butyrates and like that. So it is the glucose where from the acetyl-CoA is being produced, and the fatty acids are being produced into the milk and secreted into the milk. So the source or the raw material in non-ruminants species, it is the glucose here and there, whereas in case of the ruminants, you see good chunk is from these acetates and the butyrates. So that requires acetyl-CoA carboxylase as well as fatty acid synthesis to link two carbon chains together in case of non-ruminants. Synthesis and transfer of triglycerides within the epithelial cell begins near the smooth endoplasmic reticulum. And in the smooth endoplasmic reticulum, there is the accumulation of these triglycerides and other lipid products within the cytoplasm, results in the formation of micro droplets which coalesce into the large droplets as they translocate to the apical membrane, where they are then secreted as a membrane-bound fat droplets. That's a route what these milk fats do take from these epithelial cells into the lumen or into the milk. So you will find that here. It's a very, I mean, it will be very easy to understand over here. This is a blood capillary. You'll find everything here in the glucose and volatile fatty acids, everything, acetates, carbohydrates, and these butyrates. They will come in. This is the epithelial cell here. So this is the basement membrane of this epithelial cell. So a glucose will enter into the cell, and then you see glycerol will be formed from this. And again, you will find that fatty acids of carbon 16 to 22. You'll find this again. There's a lipoprotein here. There's a lipase, lipase over here. That's a membrane bound, and then see anything comes from here, chylomicrons or VLDL, it will be processed by LPL, and then fatty acids will be given to the epithelial cell, and here the states butyrates, the acyl-CoA, monoyl-CoA will be produced by ACC enzyme, then say into the fatty acids 4 to 16 carbons, and then this is a lipid droplet here, and this is a mammary fat globule or a milk fat globule. This is endoplasmic reticulum. It's all comes here. Acyl-CoA will be in the enzymes, and again with this, it will produce here in the endoplasmic reticulum, and ultimately being secreted as milk fat globule, which will be a membrane bound. These are other glycerol, those acyl transferases, different glycerol acyl transferases, which process the glycerol and then makes available it for the production of fatty acids as milk fat globule. Or you can see over here fat globule that's composed of triglycerides, monoglycerides, free fatty acids, phospholipids, cholesterol, and cholesterol esters, and it's being produced. Say 50% it is from fats which come through the dietary fats. They get saturated over here, and then hydrolysis in the lumen, ultimately saturated fatty acids will be coming into the intestine. They will be absorbed, and they will come into the liver, and here again in the lumen, otherwise through the luminal degradation, acetates and butyrates will be produced. They will come into the liver as acetates, butyrates, and they will be taken to the mammary gland. They will take the de novo synthesis pathway for the production of this milk fat or milk lipids, whereas those saturated fatty acids, which will be taken from the diet and absorbed intestinally, and then coming into the blood circulation, and from the blood circulation, they will ultimately reach into the mammary glands, and it will be taken up from the plasma, these fatty acids, and will be processed in the epithelial cells at the smooth endoplasmic reticulum, and ultimately secreted as fat globules, or you can say milk fat globule, and that will take the route which we have already discussed earlier. These are some of, different enzyme systems. If glucose comes in, it gets fixed into glucose 6-phosphate. Okay, it will go glycogen, biosynthesis, nucleotide biosynthesis. If it again goes fructose 6-phosphate, and then this fructose 6,1,6 biphosphate, you will find, then it will form glycerine, purines. It goes down to the pyruvates and enters into the TCA cycle. It takes that pathway, this pathway, and enters into the TCA cycle, and then forms acyl-CoA, and then comes into the lipid synthesis, and it forms the lipids in the mammary glands. Similarly, if we discuss about the milk protein, that's a casein synthesis. The milk protein we name as caseins. The source of proteins in the milk That's a casein. So milk is rich in the caseins also. A good proportion of caseins you'll find in those milk. So most of milk proteins, they are synthesized in the rough endoplasmic reticulum from the blood amino acids. So the raw material for these milk protein, that's a casein, they are the amino acids, and amino acids you'll find that they will come into the blood and to the mammary gland at the basal level, get entered into the say epithelial cells. They will reach to the endoplasmic reticulum. They will be then processed. So amino acids, they will be absorbed via basal membrane by specific transport systems. Then inside the cell, amino acids covalently bound together to form the proteins and the polysomes on rough endoplasmic reticulum. Then they get transferred from say this endoplasmic reticulum to Golgi apparatus to post-translational processing and packaging as secretory vesicles. So almost the route is same as the lactose takes through endoplasmic reticulum, through this rough endoplasmic reticulum forming those secretory vesicles. Here again, the amino acids, they coalesce together, they form, they get packaged in the Golgi and then secreted as secretory vesicles and reach to the apical membrane, fuse there and push those contents into the lumen or into the milk. So proteins synthesized include caseins, beta-lactoglobin, alpha-lactalbumin, and membrane-bound proteins or enzymes. Two major classes of proteins are casein and whey proteins, with a small amount of non-protein nitrogen substances like urea. So the casein is the major portion. In cow, casein constitutes 79%, whereas the whey proteins, they are being produced by the epithelial cells of the mammary gland itself. You won't find those in the blood. These proteins, basic proteins, caseins, and the whey proteins, which are beta-lactoglobin and the alpha-lactalbumin. So in cow, the casein will be the major portion, 79%, whereas the whey proteins, they'll be 21%. Whereas in case of the sow, you'll find that casein will be 58% and whey protein will be 42%. And in case of humans, casein will be only 35% and whey protein is the maximum 65%. And alpha-lactalbumin or you see beta-lactoglobin, the whey proteins. So this is in different animals, you'll find different composition of these proteins. See, whey proteins are packaged in the Golgi secreting vesicles and include beta-lactoglobin and alpha-lactalbumin. Immune proteins, lactoferrin, lysosomal enzymes, and the proteins and the hormones, proteinic hormones. Casein is phosphorylated in the Golgi, forming the natural organic chelation with the calcium, magnesium, zinc, iron, copper, and manganese. Alpha and beta caseins, there are different classes of alpha, beta, and kappa caseins. They contain high amounts of calcium phosphorate and link together to form the protein colloids called as casein micelles. So alpha and beta caseins, they contain high content of this calcium phosphate and it makes them to chelate and produce the casein micelles. Whereas kappa casein has a less calcium phosphate, doesn't coalesce, doesn't coagulate or coalesce together, and thus they has a this carbohydrate chain that surrounds the micelles outside and restricts from coagulating. So it surrounds the micelles, the casein micelles, through its carbohydrate chain, these kappa caseins. Inside, you'll find the alpha and beta caseins that will be surrounded by the carbohydrate chain of the kappa caseins, and thus makes these micelles, casein micelles, prevent it from coagulating together. But once you'll find that if this carbohydrate chain is digested or hydrolyzed, then there is a coagulation of these micelles and to form the clots or the curds. You'll find in case of the curds or the or the milk clots, while this protein gets coagulated, in other words, because that carbohydrate chain of kappa casein is hydrolyzed or digested, and then it makes the casein micelles to coagulate or to coalesce together and form the clots or curds. In the dietary proteins, you'll find here in the diagram, you'll find the rumen digestible protein, rumen undigestible proteins. They will be digestible proteins. They will be rapidly degraded and slowly degraded in the rumen into the amino acids or microbial proteins, ammonia. You see, ammonia and these amino acids, which is being degraded in the rumen, they will be taken by the microbial population in the rumen. This microbial population will form its own protein, what we name as microbial protein. That microbial protein will come as a source of protein in the animals, and that microbial protein will come into the intestine, and in the intestine it will get digested through the enzymatic this digestion, and once it is digested, it comes into the liver as amino acids and forms the body tissues or other milk proteins. Whereas that is coming into the feces. The other portion of this ammonia, the urea, it's being produced as a urea that excreted through the kidneys and a little bit in the blood where it maintains the pH, or it gets then secreted to saliva in case of the ruminants. Or you can find here, say, dietary crude proteins, maybe rumen digestible, rumen undigestible proteins. They will come here. If it's undigested protein, they will come into the intestine directly and get absorbed, digested, and get absorbed as amino acids. Whereas which is digestible, rumen digestible, that will be changing into the urea, or you can say into the ammonia rather. That ammonia, and they will be taken by the bacteria. They will produce its own bacterial protein. This bacterial protein will come into the intestines, and then here from amino acids. Here then urea, ammonia will come into the liver and will be changed into urea. Then urea will be again taken into the rumen itself, or maybe secreted via the saliva into the rumen. These amino acids will come into the mammary gland and will be reduced into the casein through two sources. One through de novo synthesis of those whey proteins, and then the other source from the lipids, plasma lipids, mostly saturated. These these proteins from amino acids, which will come from say besides the whey proteins. You'll find this this amino acids. They will produce the caseins in the caseins or the whey proteins, as we have already discussed about the whey proteins here. Say whey proteins, they will be in beta-globulin and alpha-albumin. So they will be produced in the Golgi secretory vesicles and packaged in the this endoplasmic reticulum. Then as the lactose is being produced, similarly this too is being produced. Another, okay, I have not seen over here. So this is how these proteins are being produced, the caseins and the whey proteins from the amino acids coming from the dietary protein. And this how these proteins are being produced. We have seen lactose being produced, seen fats being produced, we have seen proteins being produced, and similarly one specificity in the milk is that they do contain immunoglobulins that do in the early stage of lactation, what we name as the first milk or the colostrum. So that's very rich in the immunoglobulins, and these immunoglobulins are taken from the blood, and as such, because intact, they are being produced into the milk. So immunoglobulins or Igs bind to the specific receptors on the basolateral membrane of the epithelial cells, get into the cell in the endocyte or this endocytic vesicles. We have seen that pathway, that route, how they get engulfed and then membrane bound, and then coming into the cytoplasm of the epithelial cells and the endocytes, and then reach to the apical membrane, and then again through exocytosis, they push those immunoglobulins into the milk or into the lumen. So transported to the fuel with inner apical membrane and releases into the alveolar lumen as exocytosis. Then immunoglobulins are transported as such, as intact. They are not being any processed. They are being taken up from the basal membranes basolateral membranes and then taken in through those endocytic vesicles, and then on the other side of this cell, on the apical side, they are being then pushed out through exocytosis as such. No serum albumin receptors are however found. However, serum albumin may get internalized along with the Igs. You'll find some serum albumin may be there internalized with these Igs, or in some cases when these tight junctions are leaky, you'll find that serum albumin may make an entry into the milik also. Then many other, the constituents of the milik besides these major, you will have the minerals, you will have the vitamins. It's a good source of these minerals and vitamins. You'll find that microminerals, calcium, potassium, sodium, magnesium, phosphorus, chloride, and sulfur. Even the microminerals like chromium, copper, iron, iodine, fluorine, magnesium, molybdenum, selenium, cobalt, and zinc. And similarly, fat-soluble vitamins A, D, E, and K, water-soluble minerals, thiamine, riboflavin, that whole class of that B complex, and vitamin C also, and even the folates also. They are all derived from the blood. They get derived from the blood plasma and then get transported into the epithelial cells and into the say lumen or into the milik through that say paracellular pathway, or you can say directly from basal membranes to the apical membranes, membrane transport. So this is, and maybe maintained, there's a balance being maintained between milk and the blood via those active transport mechanisms between these constituents of milk. Calcium, phosphorus, magnesium are chelated to casein, since you'll find that we have found that casein is also chelated with the calcium, phosphorus, and magnesium. So good quantity of that calcium and magnesium, they get produced through casein also, while sodium chloride and potassium, they are secreted as highly available free ions. If we summarize the things over here, you find on one side blood constituents coming in from the plasma, then the secretory cell, that's the epithelial cell of the mammary gland, and then alveolus, the cavity of the lumen, where the milk is being produced, or milk is being stored and then taken out. So here you will find that water, it's coming directly into the milk, passes through the cell membrane or through those tight junctions. This intracellular transport will be there. Intercellular transports will be there. Similarly, these minerals and other things, minerals, vitamins, they will enter into the basolateral this membrane of secretory cell and will pass on into the alveolus or into the milk through this say apical membrane directly. Even immunoglobulins, we have discussed that they will take a route there and forming those vesicles, but as such they will not be converted anything anywhere, but as such will be taken from here as secretory endocytotic vesicles and then through exocytosis into the blood. Then amino acids from the lipids, they will come inside, and amino acids, the protein source, they will be processed. They will be processed and then they will be manufactured as beta-lactalbumin or alpha-lactalbumin, what we name as whey proteins, and then either caseins, alpha, beta, and kappa caseins, and will be secreted. It will be processed there in the epithelial cells, being secreted there, being produced there, and then come into the say alveolar cavity, into the lumen. And similarly, if glucose comes into there, one is the energy source for the mammary epithelial cells, and then glucose will be then fixed. It will come to the galactose through that uridine diphosphate taken into the Golgi, and then glucose again there in the Golgi, and then it will form the lactose under the that enzyme system, lactase, which is having two components, which I discussed there, and this lactose once formed there will be packaged into the endoplasmic reticulum, Golgis, and will be taken as secretory vesicles to the apical membrane, and from the apical membrane it will be exocytosis will be there, and it will be coming into the say here into the milk, milk. And then similarly, the acetates, butyrates, or the fatty acids, they will come into this epithelial cell, will enter into the epithelial cell from the blood plasma, and it will make the triglycerides. Again, they will get processed in their acetates and butyrates. I already discussed that these will be producing those de novo synthesis of lipids, and ultimately those short chain fatty acids, long chain fatty acids, triglycerides, and then they will take the root of that milk fat globule root with a membrane, and they will be secreted into milk as globules, as droplets, milk fat droplets, globules. And this is the whole concept of this milk synthesis of different components. And if you see the composition of milk in different species, you'll find it will be given in the books. But you will find here, you see the lactose, which is not varying much in the animals, particularly in the domestic animals. Over here, you'll find it will be a major in case of, say, humans. Here you'll find here, say, cow, dog, and you'll find here roughly 5 or 4, and it will be very less in these mammalian, these say sea animals. You'll find in goat 7.4, 4.8, like that. It's not much variable there. Whereas you'll find other casein and whey proteins, you find again a good quantity of it is fats will be varying very much in different species. And in fats, we find that fats will be very high in case of those sea animals. Sea dolphin 33%. If I find in Chinia, in elephants, even much more in sea over here, seals or forest. That's again the sea animals, very rich in the fats. So this is ash and this is energy you find in. And similarly, you'll find the colostrum, very high quantity of this solid as lactose, little, but fats, good, proteins, very rich. And because it has a good quantity of immunoglobulins over here, then casein is almost doesn't vary much. So this is about the composition of milk in different species. You can consult the books, you can see there in different species how these proteins, fats, and lactose, or you can say the carbohydrates, vary in different animals. This will be all for today's lecture, and we'll be meeting again with one more lecture. Till then, I take leave over here. Thank you. Thanks a lot.